Have you ever wondered what would happen if you took 10 million aggressive human brain cancer cells and left them alone in a small dish for a week? Well, I tried exactly that. And what happened was far more bizarre than I expected. Today, I invite you to join me on this little microscopy journey. So, let's explore it together. In this video, you'll see how independent cancer cells assemble into strange, giant moving clusters. Watch as these clusters crawl across the plate, colliding, merging, and forming even larger collectives. Some cells break free from the cluster, fleeing the swarm, while others rush in, drawn into the growing biomass. You'll see what happens inside the cluster and around it. And you will also see giant clusters of rivalry in a literal tug of war, pulling neighboring cells to their side like competing empires. In the last video on scratch-assay and regeneration, I observed an unexpected phenomenon. Cells started to form these weird structures. Initially, I thought this was some random debris, clumps of accidental dead cells. However, after a little observation, I realized it's very far from the truth. These structures are organized and alive. So today, I will replicate conditions that we had last time, only this time we will have an enormous, crazy amount of cells in the plate, to give them a freedom to play. Here it is, 10 million aggressive cancer cells in this tube, like from some sci-fi horror movie. Except instead of New York, the invasion starts in this dish. You know that scene where aliens merge into one giant monster? You are about to see a cell version of it. I will transfer them to this plate and put them under the microscope. Set the temperature to 37, basically a luxury spa for cells, with a steady flow of necessary gases. And then I will leave them alone for a whole week. What happens next is entirely up to them. Here are the very first moments after seeding. Each bright little sphere you see is a single cell. Right now, they're still floating freely, with no sense of direction, just drifting along the invisible currents of the liquid. Let's zoom in a little closer. You can see some cells extending and retracting fine projections, as if they're trying to catch hold of something. They're probing, searching for a surface to anchor to. Others just drift and bump into one another, looking almost clumsy in the process. Now, let's zoom in with a stronger lens. At this stage, most of the cells are still round, their natural shape when floating freely. The moment a cell makes contact with the surface, it begins to change dramatically. Take this cell here as an example. First, it drifts down, round and compact, then it releases exploratory extensions, sensing ground for grip points, nutrients, neighboring cells, and other chemical cues around. Specialized adhesion proteins allow it to stick, and the cell's inner framework rearranges until finally it reshapes into a wide, flattened form, stretching itself across the surface, like claiming territory. It's the shift from passive drifting to active anchoring and exploration, as you are about to see now. You can see more and more cells undergoing this same transformation. A large fraction have already landed, spreading and starting to interact, laying the groundwork for a future collective performance no single cell could achieve alone. Now, let's pay attention to this one. It's much larger than its neighbors, several times the size. Maybe it carries a rare mutation, maybe something unusual in its regulation. Whatever the reason, it looks strikingly different. We will see if it also behaves differently from the rest.
As days pass, the cells have established many individual communications. However, what you see now starts to happen on a much larger scale. It looks as if entire groups of cells are building vast connection bridges across the whole plate. Structures made not of one or two cells, but dozens working together. And then something mesmerizing happens. These networks begin to reorganize. Masses of cells stretch and assemble into giant living collectives. And they don't just sit still. These clusters begin to move. Slowly at first, they start roaming across the plate. It's a breathtaking sight. Cells that were once separate and independent, now acting together like one coordinated entity. While the clusters roam across the plate, let's return to that giant cell we saw earlier. From above, a massive cluster advances to it, pulling in every cell it touches, making them part of itself. Look at the trail it leaves behind, a field that is like clear of life.
The question is, will our giant cell resist this living tide, or be absorbed into it? Against all odds, it manages to hold its ground, living on its own terms.
Now, let's watch the same scene again, but zoomed in, so we can see in detail how these clusters emerge and behave. At first, you see vast bridges stretching across the plate, linking distant group of cells. Then, cells begin to gather into specific spots, forming star-shaped hubs that slowly condense into almost perfect spheres. The birth of a collective. These clusters don't just stay still, they crawl. And occasionally collide. Merging into even larger structures. And wherever a cluster passes, it leaves behind an empty field, a void of life. But what's truly remarkable is how quickly lone, independent cells rush in to reclaim that space, feeling it as if nothing had ever been lost. Look closely at this spot. A handful of cells sit on the border between two great empires, each cluster pulling, determined to absorb them. A living tug of war unfolds before our eyes, and in the end, the cluster on the left emerges victorious, dragging its prize away. Look up. The emptied space is filled. Independent cells spread back in, leaving no gaps behind. Let's zoom in on one of these spaces with a stronger lens. Here, each cell reveals itself in full detail. Pay attention to this cell. You can clearly see the nucleus, command center, packed with chromosomes that store all instructions for life. In the surrounding cytoplasm, the scene is nothing less amazing. Tiny vesicles, little transport bubbles, are darting everywhere. Some deliver nutrients, others recycle waste, some even carry chemical messages. It's like rush hour traffic in a microscopic city. Now look at this cell here. It throws out a long, thin extension stretching across the entire field of view. It is as if the cell is laying a cable to its neighbors, or maybe just saying, hey, anyone wanna join my future cluster? Remember, these are the very same cancer cells that just assemble into those massive migrating clusters. But at this scale, you can appreciate how much coordination and restless activity is happening inside each individual cell. The distinct processes that together can fuel the grander collective. Let's compare now how cells look at the start, middle, and the end of the cluster's formation. It's very spectacular. First, floating cells, then roaming adherent cells, and finally, clusters. Now look at these clusters. They're no longer just tiny little cell gatherings. These are gigantic collectives made of dozens, maybe even hundreds of cells packed together. They're so large, you don't even need a microscope to see them anymore. Just look, I'm filming them directly with my phone. See these whitey spheres? That's not dust, those are clusters. And honestly, they look so enormous. And then, a new idea came to my mind. What if I take just the clusters, scoop them out, and seed them onto a fresh dish alone? Will they stay together? Will they fall apart? Let's see. Here comes my advanced scientific instrument, a spoon. Okay, technically it's a spatula, but today it's a cluster scooping device. With this high-tech tool, I'm going to carefully collect these cuties and transfer them to their brand new plate. Here we start a new experiment. Once transferred, they float a little, but soon they stabilize and land. What's fascinating is that in this fresh environment, where there is plenty of open available space, clusters begin to fall apart. Cells start to abandon clusters, starting a new independent journey. For example, watch this. Right in the center, an adventurous single cell decides to go rogue. It wanders away across great distances, boldly exploring new territory, while some others stay busy forming new extensions, weaving fresh connection networks. And look here, next to this massive cluster is a giant cell that has broken free. It looks enormous, almost majestic, compared to its siblings. Maybe it's that giant from the past that eventually got swallowed. Now he's taking his opportunity to get free again. Right next to our rebel, there is a cell with a perfect neuron cosplay. Star-shaped extensions sticking out everywhere, like it's auditioning for a neuroscience textbook. As you can see, cells have almost covered the entire surface of the plate again. And when everything seems to get very quiet, look at this madness that is about to happen. Giant cluster turns back and starts chasing the escapee. What is going to happen? Well, before we proceed watching it, let's rewind a little bit and watch how the great escape even happened at the start. First, a few smaller cells slip away quietly. Suddenly, look at this. A huge extension bursts out of the cluster. A dramatic push for independence.
Me when group project starts. Me killing it with Katy Perry and DJ switches to BTS. Accidentally leaves the door open. My cat with unlimited food and affection.
Watch how gracefully it unfolds, almost like dancing in a translucent tulle dress, giving the most elegant performance. And the microscope field becomes its ballroom. But this freedom doesn't last forever. The cluster soon turns back for what it lost. Switching to a stronger lens, we can see the details. This cell is so gigantic that even its nucleus is larger than most of its neighbors. Its membrane ripples with delicate ruffling at the edges, breathtakingly beautiful, but not for long, as the cluster begins to swallow it back. And since we are on the cluster now, let's explore it in more detail. We'll attempt to 3D scan it, slicing through its layers from top to bottom. At the very top, things look calm, but wait until we dive deeper. Here, in the middle, there are plenty of gorgeous cell extensions that remind me of hydras. We will get back to them later to see what they do. Moving deeper. Now, we can see them clearly. Dozens of spherical units packed together. These are the individual cells that make up the cluster. Look how many there are, each one contributing to this living collective. And at the very bottom, notice how the cells are stretched, anchoring and pulling the whole structure together. Magnificent! Now let's focus back on those peculiar extensions in the middle layer. They look like hydras that sense and capture nutrients from around. The cluster seems like making these surface cells to specialize on it, while the bottom ones pull the whole structure forward. Magnificent! However, seemingly not for everyone. Look at this funny looking cell that is attached to the external area. It seems like it's trying to move out. Then pulling its friends along. Eventually, it steals around 6 cells from the cluster with it. That is so hilarious. Let's look at this again. Alright, Operation Escape the Cluster has begun. One rogue cell currently captured by the cluster. However, it did not abandon its connection to the plate. Watch as it pushes itself along the fiber, Philopodia, its very own grappling rope, and starts to crawl away in full stealth mode. Every tiny move is precise. The cell is basically Tom Cruise in a petri dish.
Alright, now look at this maniac right here. Look at this crazy play of extensions, mesmerizingly beautiful. Not only is he the biggest, but also the most hardworking cell in the cluster. Everyone else is chilling in comparison to him, when this one is out here speedrunning cell behavior like it's getting graded. I don't think I've ever seen such behavior in single cancer cells before. Whatever he is doing here, he's doing it at 200% efficiency. I can only hope the cluster has some bonus system and this dude winning Employee of the Month. Okay, gorgeous! Now let's zoom back again and see what happens to that cute giant cell escaping. The cluster swallows part of it, dragging downwards, but the giant cell resists, crawling upward with a grace spanning through the great distances. How is it going to end up like? Well, it's really stretching. Well, until it eventually completely disconnects. Well, that was exciting. But while our eyes were locked on the drama, the rest of the plate had transformed. The space that was once open is now filled edge to edge with cells. They have woven themselves into a continuous network, stretching in every direction. A web of living connections across the entire plate. Wow, look at this absolute masterpiece. The cells have conquered the entire plate, a living carpet of activity, shimmering with motion. And there, scattered like tiny kingdoms, a few remaining clusters hold their ground. Wait, hold on. Hey, these two giant clusters look like they're about to merge. Let's zoom in. This could be legendary. Oh, come on. The bottom one just turned around. It's like, nope, not today, buddy. No content for you. I'm heading the other way. Classic. You spend days setting up the experiment and the cells just fail you. But, okay, that's fine. Let's at least take a closer look at this one. What a mesmerizing sight. The edge is alive with constant motion. Tiny vesicles bursting, apoptotic bubbles drifting, shimmering details everywhere. It's chaos, but an organized, beautiful kind of chaos. A dance of a million molecules, all alive, all in motion, truly mesmerizing. But not all stories end up in a triumph. After a week of watching these cells grow, merge and move like living cities, silence fell. Every single one of them stopped. At first, I thought it was just part of the experiment. Maybe nutrient exhaustion, maybe internal collapse. But then, I found the real reason. One of the hoses feeding the CO2 and other hydrated gases had been slowly colonized by bacteria. Or maybe it's a fungi. A tiny biofilm had grown inside, blocking the flow, suffocating the entire plate. Ironically, while studying one form of life, another crept in and ended it. But that's science. So, in the next video, we are going to follow the invade itself. We will isolate it, culture it, and see who is the saboteur itself. Thank you so much for watching it. It took me more than 3 months